Hi, my name is Mr. Baker, and welcome to another thrilling day of science. I am here at home with my teddy bear. This is the tame teddy bear that will be bungee jumping with me today. Say hello to him. Thank you. And I figured I'd start out with him a little bit. I hope everybody's good there. I hope everybody's willing to and ready to pay attention and learn a lot today. I am going to be going over quite a bit of stuff, so make sure that you are completely with me. You're going to want to be, all right? So I'm going to put aside Teddy, and we're going to get going. First, turn your pages open to open your book to section 3.5. Hopefully everybody has their book. If you need to borrow one, you have about 10 seconds in order to go over to my corner where my desk is onto the windowsill and pick one up. So I'll just give you a second there, and I'm gonna figure that you're just about done right now. So let's get going. Every once in a while you're going to lose me, you're not going to be able to see me, and that's fine. That doesn't mean I'm not watching you, it just means that you can't see me. I hope you understand. Section 3.5 says, explain the structure and function of the chromosomes found in the nucleus. And that's what we're going to be doing. So the structure and function, that is basically what, that is what, chromosomes look like and what do they do. So right here, this is DNA. DNA is found in the nucleus of your cells. This is DNA magnified a lot. It looks like a spiral staircase. Or imagine a twisted rope ladder. So DNA looks like a spiral staircase or maybe a rope ladder that has been twisted. Imagine that this is something that you just kind of take. That's a rope ladder, and you've taken it and you've just sort of twisted it, so that you can, like this, so that it's all tangled and spiraled like that. Actually, technically, every rung or step of the ladder is a piece of DNA. So if I was to draw this real quick, it would be like this. Yeah. So this would be DNA, and then this next one would be another piece of DNA, another piece of DNA, another DNA, and actually, technically, you can see how the DNA turned color in between. Technically, this is a strand of DNA, and then this is a strand of DNA, but we're going to call this whole thing one DNA. So if I was a count, I'd say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, let's say there's like 25 pieces of DNA to make up this whole strand. So this whole thing here is about 25. Now, this doesn't seem like an important number, but the fact is these numbers I'm going to be throwing out are kind of important. 25 here. Now, many of these strung together make a gene. Now, I've got 25 here. When I say many, I'm talking thousands. Okay, so figure on an average 1,000 of these strung together will make up a gene. The average gene is made up of, let's say, about 1,000 DNA in a row. So if DNA were actually this size, what you're seeing is pretty much, I don't know, maybe 2 feet long or so on the screen, on the board. So 2 feet long is 25. So if we're talking 1,000, that's like 40 times that. So that's 80 feet. Imagine that this DNA strand reaches from our windowsill all the way to the auditorium. That is one gene. If DNA were the size of what you see on the board, one gene would be about 80 feet long. Okay, so that gives you an idea of what a gene is made up of. 1,000 DNA. And those 1,000 DNA come together to make up just one gene in your body. Now, of course, your body has a ton of genes. Your body has genes for your hair and your eye color, genes for how tall you will be, and and genes for, you know, how muscular you will be, and genes for your personality, and genes for everything that goes on in your body. Pretty much. What that means is that you've got a lot of genes. So each gene is made up of, let's say, 1,000 DNA. But down here, not only do many genes, many DNA make up a gene, but also, you're going to have to deal with my slowness here because I'm capturing at the same time and then I'm drawing and doesn't like it. But many genes strung together make up what we call a chromosome. So, if DNA, once again, were this size, you know, the size of what you see on the board, one gene would stretch 80 feet. Many genes strung together. When I say many, I mean thousands once again. Thousands. Thousands of genes strung together make up a chromosome. So, getting back one more time. If genes are the size that are on your board, one gene would stretch to the auditorium. But a whole chromosome made of 1,000 genes on the average, well, that's not 80 feet anymore. That's 80,000 feet. Divided by 5, that's 16 miles. So figure that a chromosome made of DNA, the size of what you see on the board, which stretch from here to basically like the center of Cheshire, okay? Or probably farther, from here to New Haven, okay? About 16 miles. That's a big chromosome, okay? But of course, it's made of DNA that are, you know, that are big. Your DNA are very, very small. I mean, they're molecule size, almost atom sized, and so you can fit all these chromosomes in your nucleus. Now, to go over, many DNA, thousands, make up a gene. Many genes make up a chromosome. Think of it this way. It's like a train, okay? A train, as you might be able to see, is made up of these little slats of wood. Let's see if I could make a nice color for the slats of wood. Blue! So you see this slat of wood here, this little piece of wood here, and this little piece of wood here, and this one here, and so on and so on. Each one of these slats is a DNA. So you've got many of those slats, boom, 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 and they make up the car. And many cars, if you've ever sat at a train crossing, while the train goes by, and you gotta wait and wait because there's hundreds of cars that go by, right? Maybe even 1,000 cars go by. That whole train is the chromosome. So the train is made, so you can't see my hands, but they're way out here. The train is made of cars, 1,000 of them. And each car, imagine being made of 1,000 of those slats of wood. Train made of cars, cars made of wood. That's the same idea as saying a chromosome is made of 1,000 genes, and the genes are made each of 1,000 little DNA. So DNA make up a gene, and genes make up a chromosome. So just as a train is made of cars, which are made of wood, you can think of it as, come on, oh, bugger. Just like a train is made of cars, and cars are made of wood, same thing as a chromosome is made of genes, and the genes are made of DNA. Okay? Now, In each one of your cells, you have 26 pairs of DNA. I said that wrong. In each one of your cells, you have 23 pairs of DNA, which makes 46 total. So let me make this easy here. Or maybe it won't make it easy, I don't know. Okay, so there's 26 pairs of chromosomes. And that equals, I said it wrong again. I got 26 on my head. There's 23 pairs of chromosomes, which makes 46 chromosomes total in each cell. Okay, that says each cell right in there. Okay, let me move this up so you can see it. Hello, I don't want that. Come here. That's right. Let's move this so you can see it. Okay, 23 pairs of chromosomes. What? Come here. Everything's slow and weird because I'm recording at the same time. So 46 chromosomes total in each cell. Each one of those chromosomes is made of 1,000 genes. And each gene is made of 1,000 DNA, which means there's a million, there's a million DNA in each chromosome. 1,000 times 1,000. Which means that there's 46 million DNA in one nucleus of one of your cells, which is pretty amazing. Now, of course, remember, you have 50, on the average, 50 trillion cells in your body. So now let's quickly do the math. 46 million DNA in one cell, 50 trillion cells in one body. That's a lot of DNA. That's 50 trillion times 46 million. We're talking like 225 blobillion DNA just in your body. These are some big numbers. You can do it out later. I made up blobillion. There's no such thing. You don't have to know those numbers. I just want to give you an idea of how incredibly complicated your body really is. It's pretty complicated. Now, what do chromosomes do? Okay, it's complicated. This is what I want you to know. I want you to know that a chromosome, remember when I said a chromosome is like a train, and the train and the genes are like the cars on the train, and the wood is like the DNA that make up the genes? The wood that make the cars is like the DNA that makes the genes. Now here's another way of looking at it. A chromosome is really not like a train. It doesn't act like a train. What it really acts like is a cookbook. A chromosome is like a cookbook. Now, of course, when you have a cookbook, you know what's inside a cookbook. You open any page, every page has a different recipe, right? And each recipe all together make up a cookbook. So now imagine a cookbook with 1,000 recipes. It's not hard to imagine. You know, recipe on this page, recipe on this page. You got, all the pages. That is a pretty thick cookbook, 1,000 recipes. But that's what a chromosome is like. It's a cookbook of recipes. Each recipe is a gene. And what are you doing to me? Oh boy. All right, whatever. Oh my God, where is it going? Each recipe is a gene. Okay? So on each page of the cookbook, there's a recipe. Just like in each section of a chromosome, it's a gene. Now, genes can be broken down into DNA, just like a recipe can be broken down into each and every word. the words in the recipe, I did it again. The words in the recipe are like the DNA in a protein. The words in the recipe are like the DNA in a gene. And that's exactly how it works. Because each gene is the recipe for making one protein. Now, you know how your genes make your eyes blue or brown? That's because that gene that makes your eyes blue or brown makes a protein that gets shown in your eye as either blue or brown. Technically, actually, that gene, blue is the absence of the protein. So that gene, that if you have a blue-eyed gene, it just means it doesn't make the protein. If you have a brown-eyed gene, it does make the protein. You don't have to know this. But the point is that genes are the recipe for proteins. If a gene is needed to give a recipe to make a protein, it will give its recipe to the cell. And then the cell will make that protein. And then that protein ends up making you, because cells are made of protein. Okay. So, words make up a recipe. Recipes make up the cookbook. That's like saying DNA, which are the words, make up a gene, which is the recipe. Recipe, which is the gene, or gene, which is the recipe, make up a chromosome, which is the cookbook. And those four, whether the 23,000 pairs of genes all go together to make you. You are basically 23,000 recipes of proteins that all come together to make you. You're made out of 23,000 recipes. I mean, 23,000 pairs of recipes, really. Okay. Let's go down. I can go down without losing me, huh? All right. So here we go. When a cell needs a protein, this is what happens. I'm going to extend the page, because I'm going to have to draw this, and it's not going to be pretty. When a cell needs a protein, this is what happens. The gene, which is the recipe, in the nucleus gets read. When a cell needs a protein, the gene in the nucleus gets read. And I'm going to show you how it gets read. And a copy of the recipe goes out of the nucleus to the ribosome. Remember, everything's kept in the nucleus. All these genes and chromosomes and DNA and all that, I mean, you know, the DNA which makes the gene, which makes the, this is all found in the nucleus. But you see, inside the nucleus, they don't make protein. Proteins are made at the ribosome, which is outside the nucleus. So somehow you got to get the message, the recipe, from inside to outside. So let's draw this so far and see what's going on. Come on. I'm going to draw it using this because I'm very smart. Here is the nucleus. Very slowly. Okay? Now notice I draw it with a dotted line, because what this is right here is the nuclear membrane. So inside here is the nucleus. And for inside the nucleus. Okay? Out here is the rest of the cell. Imagine that the nucleus kind of goes. I didn't draw the whole thing. Okay? So, like, the cell is even bigger than this whole thing. So I'm just drawing this one area of the outside of the nucleus. I mean, it goes all the way down and around and comes all the way around here. So I only drew kind of one small piece, magnified very big. So this nucleus is surrounded by this skin called the nuclear membrane. notice that there's holes in the nuclear membrane for things to go through. Inside the nucleus, there are chromosomes. So here's a chromosome. So that's a chromosome. If I was to draw one little gene from the chromosome from here to here, that's one little gene in this chromosome, 1 out of 1,000 genes. I'm going to make this bigger so that you can see it. This is me telescoping this gene out so that you can see it. So this one gene I'm making a bracket to show that this is all one gene. I haven't drawn the gene yet, of course. Here, let me draw the gene in a different color, just because I feel like it. Here's the gene. You'll see that it's bigger and fatter than before. A lot of drawing in this. Okay, now remember from that first picture up there, all the little parts, all the little rungs to the ladder are the DNA. So now I'm drawing the DNA. Drawing the DNA. So this is all the DNA that make up this one gene that's right here. It's very small on the chromosome, but now we're making it magnified. So here are all the DNA. This one gene is actually the recipe for one protein. What happens is there's these little messengers, and they go into and out of the nucleus. So I'll draw a little messenger. I'm not going to go too crazy about this. Basically what happens is this gene actually has a zipper down the middle. Okay? The zipper unzips, and this little messenger, I'm just going to call it a messenger. This little messenger, M for messenger, goes in. Should have made it a different. Goes in. Once it unzips, I can't draw it unzipped, but assume that this zipper is open like a fly on the pants. And this messenger comes in and actually reads the gene once it unzips. And then it leaves and goes out this way, out through one of the holes and out to a ribosome. So I'll make ribosome a different color, green. So here's the ribosome. Ribosome. Now this messenger has the recipe for how to make the protein, has read it, now leaves the nucleus and comes on out to the ribosome. Here it comes. And gives the recipe to the ribosome. All right? The ribosome takes it and basically makes a protein out of it. All around the ribosome are little amino acids. They just are floating in the cell. They're just floating around. A. And the amino acids come to the ribosome, and depending on the recipe, the ribosome grabs certain amino acids and puts them together, and the amino acids get put together with another amino acid by the ribosome. The ribosome is kind of putting it together, is making the recipe. It's kind of the kitchen where the recipe gets made. So these amino acids are starting to connect together, and ultimately, when the whole recipe has been read, all these amino acids connected together like pearls on a pearl necklace. This thing here is the protein. All right, at this point, write down some questions if you have any questions, because I'm sure you've got questions like, I don't know what you're talking about, or how does this really happen? It feels like you're leaving some big important steps out, which I am. I'm leaving out big important steps. I mean, this messenger that goes in and reads, there's actually several messengers. One messenger for each DNA that go in and read. And then those messengers connect, and then they go out to the ribosome. But we'll deal with this later. It's good enough to know that there's a messenger that goes in, reads the gene, reads the recipe in the nucleus, and then leaves and goes back out to the ribosome and tells the ribosome how to make this protein. Now that this protein is made, it can go through the endoplasmic reticulum to wherever it's needed. Let's say, I don't know, there's a part of the cell membrane that's broken. So somehow that message gets into the nucleus. The nucleus unzips the right, this is how it actually, unzipping is actually a real term for this. The nucleus will unzip, the little messengers come in and read the unzipped portion of that gene. That whole gene unzips, reads it, and then it leaves and goes out to the ribosome, where the protein that will repair the cell membrane gets made. So that protein gets made, and that protein gets out through the endoplasmic reticulum to the cell membrane and repairs the cell membrane. Pretty cool. And that is the whole point of genes and DNA. They provide the recipes for a cell to make itself. And in turn, all the cells that are made make up you. And that's why you are a product of your genes. You're a product of all the recipes that are in your body, that are on all these chromosomes that are in the nucleus. Any questions? Write them down. Haha, you can't ask me. Next. Let's go to the next page. V6. All right. 3.6. Describe how the cell membrane helps the cell to maintain a balance between the internal and external environments of the cell. Osmosis. In other words, what is osmosis? Come on. Bang. What is osmosis, and how does it help the cell? All right. This has nothing to do with chromosomes or genes or the nucleus or DNA or anything like that. This is completely different section 3.6. All right. What is osmosis? How does it help the cell? First, in order to understand osmosis, we should first learn what diffusion is. Diffusion. Diffusion, yeah, you stink. Does that every time. All right, diffusion is when anything moves from an area of greater concentration to an area of lesser concentration. Okay. This only makes sense. I think a while ago, well, maybe I didn't mention this. Imagine this. I take all of you, and I tell you to stand up, and all of you cram yourselves into a corner of the room. Like that one over there. Or, no, actually, not that one. That one over there, okay? Everyone go cram yourselves into that corner where, you know, farthest away from me, closest to the window, over there. And so you all cram it. Don't do it. So you all cram into there, let's say. You all cram into there, and I say, okay, I'm leaving. I'm going to go and get a coffee. Not that I drink coffee. I'm going to go and play video games in the teacher's lounge. All right? So I leave. I say, I'm going to be back. I don't know when. I leave. When I come back, let's say it's 5-10 minutes later, are you all going to be in the corner anymore? No, of course not. You're going to stand there for a little while, but eventually you're going to be like, you know what? I don't want to be in the corner with everybody anymore. I want to go to where there's fewer people because I'm crammed in here and I don't like it anymore. So you're going to move to where there's fewer people. That's diffusion. You're going from an area of greater concentration, all of you crammed in the corner, you're all concentrated there, to an area of lesser concentration, the rest of the room. So you diffuse into the rest of the room. And that is diffusion. Diffusion happens all over the place. If anyone's ever put plastic wrap on a bowl of, I don't know, tuna salad or something like that, and then you go up and you smell, can you smell the tuna salad through the plastic wrap? Yeah, you can. You can smell the tuna. But wait, there's plastic wrap over it. Shouldn't the plastic stop all the smell stuff? No, it doesn't. Because once you put the plastic wrap on, you're concentrating all the smells inside that Tupperware, or whatever the heck it is. It's a bowl of tuna salad. So all this tuna smell is concentrated in there. And what happens is it finds little tiny places to diffuse through the plastic wrap and out into the outside where there's less tuna smell. If there was more tuna smell out here, it would diffuse into where there's less tuna smell. If there was, I don't know why there would be more tuna smell out here than in where there's tuna, but if there was, it would go the opposite direction. So it will always go to where it's less concentrated. If you're cooking in the kitchen, all right, let's say you're baking an apple pie. There's an apple pie being baked in the kitchen where you live, okay? The smell of the apple pie doesn't just stay in the kitchen. It doesn't stay concentrated in the kitchen. It travels all throughout the room, all throughout the house or the apartment, and it gets into all different places. That's diffusion. Diffusion is a sort of a fact of nature. It's a universal thing. It always happens. You put your clothes away neatly in your bureau or your drawers or whatever. And it's because you were told to clean your room. So you clean your room. You put everything away, put everything away, put everything away, everything away. Your clothes get put away, everything. Toys get put away. Everything gets put away. And in a week, where is everything? It's back diffused out of where it was concentrated in your drawers. Like, that's where your clothes are concentrated, in your bureau or whatever, wherever you keep your clothes. The high concentration of clothes, it just naturally diffuses out onto the floor where there's less of a concentration of clothes. This is actually something that happens very naturally. It's called entropy. Entropy is the tendency for all things in the universe to become less concentrated, less organized in one place. So the next time when somebody looks in your room and says, "How can you? Why is it like this after only a week?" You say, "Well, it's only operating according to the laws of the universe. It's supposed to become less organized, less concentrated. It's supposed to spread itself out. That's diffusion." Try that. Try that line the next time you get called on the fact that you have a dirty room. Anyway, all of that has to do with diffusion, which is when stuff goes from where it's very highly concentrated in one area and spreads to where it's not highly concentrated. Spreads out, and eventually it'll spread out so that everything is equally concentrated everywhere. Just like if you put a drop of food coloring into some water, does the food coloring drop stay a drop? No, it diffuses out, and eventually all of the water inside that container becomes the same color. It all perfectly diffuses so that everything is the same concentration. Now, what does this have to do with a cell, right? That's what you want to know. Well, diffusion usually, and especially in a cell, diffusion usually happens through a membrane, like a cell membrane. Diffusion allows all the good stuff to go into a cell and all the bad stuff to leave. If all the good stuff is concentrated outside of a cell, well, it goes from greater concentration outside of the cell to lesser concentration. It'll come into the cell. If bad stuff like waste is highly concentrated in a cell, well, it'll go from highly concentrated in the cell, and it'll leave and go outside the cell. Osmosis, now, that was the important word, right? Osmosis. Oh, stop it. Osmosis is a type of diffusion. It's a special type of diffusion that only has to do with water. Osmosis is really just a special word for water diffusion. It's when water enters and leaves the cell. It's really just water diffusion. Everything else is called diffusion. When it's water, it's called osmosis. Okay? So when water goes from higher concentration of water to less concentration of water, it's osmosis. Anything else is called diffusion. Don't ask me why water has a special word for its own diffusion. I don't know. Okay? But water being very important, it does have its own name, and so let's look at some osmosis of water. Osmosis happens with water so that the environment in the cell is about the same as the environment outside of it. All right, so think of a cell's insides as lemonade from a mix. Okay. You ever make lemonade from a mix? You know, you scoop it out of the tin, you put some into your glass, and then you put the water into your glass, and then you mix it up, and you drink it, and it's delicious. It's delicious because it's lemonade. Inside that lemonade that you made, the little lemonade particles, okay, are swimming around in the water. Okay? Think about all the little things in a cell as the little particles of lemonade. Okay? A cell has water, and a cell has particles inside. There might be sugars floating around, or amino acids floating around, or all sorts of other stuff. And they're floating around in the cytoplasm, which is basically water. So, take a look at this picture. The dots are the particles. Where you don't see a dot, that's water. Okay? Out here, the dots are particles. Everything that isn't a dot is water. Now let me ask you. In this first cell on the left, where is the water highly concentrated? Where is there a ton of water in a given area? All right. You're all thinking, maybe you're even answering, I don't even know what the heck you're doing. But the fact is that in here, where there's a ton of particles, there isn't as much water. It's mostly particles and only some water inside this cell. Out here, there's not as many particles, which means that the concentration of water is higher out here. Less particles, more water. Inside here, more particles, less water. Osmosis always happens from where water is greater concentration to where water is lesser concentration. So which way water will go is, in this case, on the left-hand side, oh, stop it. Water will go from where it's greater concentrated into the cell to where it's lesser concentrated. In this case, water will want to come into the cell. Think of this cell as lemonade. This cell is going to be really strong lemonade. Look at all the particles of lemonade. This cell is going to be strong lemonade if this cell was, if you think of these particles as lemonade. Really strong lemonade. And outside the cell, just a few little lemonade particles, but mostly water. This is going to be really weak lemonade. So which direction are you going to move water to make the lemonades about the same? Right? which way are you going to move the water so you make the lemonade about the same? You're going to move, I should say the water. So the lemonades are about the same. You're going to want to move the water into the strong lemonade. That will weaken this strong lemonade inside the cell and make it more like the outside. So, in this first situation, which direction will osmosis occur? Osmosis will occur into the cell. Here's the opposite situation. This cell has not a lot of particles inside of it, and it's surrounded by lots and lots of particles. It is highly concentrated in particles out here, but not highly concentrated in water. More particles, less water. Inside the cell here, less particles, more concentration of water. So water going from high concentration to low concentration. Where is it highly concentrated? It is highly concentrated right smack dab. It is highly concentrated in the cell. The water is concentrated in the cell, and this highly concentrated water will move out of the cell. Always think of it this way. If these particles are lemonade, you're going to want to add lemonade to where it's strong to weaken it out and make it the same as the other area. So where is there strong lemonade? Outside the cell in this situation. Where would the water move? To the strong lemonade to weaken it out so that it can be the same as in the cell. And that's about it. I'm going to stop here and hope that that made sense. I will see you guys on Monday. If there's any time left, I want you to relax, take a deep breath, stay in your seats, yap a little bit to your neighbors, not across the room. I hope you have been good to me while I've been talking to you and to your substitute. And we'll go over this on Monday. There is no homework. We will not have a test on Monday. I'll make sure that you guys understand it on that day, and there'll probably be a quiz on Tuesday, okay? So, enjoy your weekend. I will see you soon. Have a good rest of the day, and bye. And Teddy says bye too.